أسعد الله أوقاتكم بكل خير أحبتي، والسلام عليكم ورحمة الله وبركاته. معكم الأستاذ المساعد الدكتورة مروة حميد الخفاجي، دكتوراه في الأحياء المجهرية، قسم علوم الحياة، كلية العلوم، جامعة بغداد. هذا المقرر Food Microbiology، علم الأحياء المجهرية في الأغذية، لطلبة المرحلة الرابعة للدراستين الصباحية والمسائية. المحاضرة الأولى بعنوان العلاقة بين الأحياء المجهرية والأغذية والفروع الجديدة من علم الأحياء المجهرية في الأغذية. The relationship between food and microorganisms and the new branches of food microbiology. One of the most important discoveries in history of biology occurred in 1665 with the help of a simple microscope. Antony van Leeuwenhoek discovered the world of microorganisms. When he made this simple microscope, he discovered the smallest structure units, called them animalcules. He observed them in food residues obtained from his teeth, in juice, and in vinegar, and raindrops. And this was the first report about the relationship between food and microorganisms. The figure shows A replica of a single-lens microscope by Antony van Leeuwenhoek, and B, air-dried smear of blood through the original van Leeuwenhoek microscope. Many scientists and philosophers believed that some forms of life could arise spontaneously from non-living matters, and they called this process spontaneous generation theory, SGT. They believed that snakes and mice could be born from moist soil, fleas from manure, maggots, fly larvae, worms, microorganisms from spoiled meat and food. As we see, living creatures from non-living matter. They call it spontaneous generation theory or abiogenesis. In order to refute such theory, Francisco Redi filled 3 flasks with meat, one left open, the second sealed, while the third covered with a piece of gauze. He noticed that maggots appeared only in the open flask after fleas entered the flask and laid their eggs, but the other flasks show no maggots. However, a putrid flavor appeared in the sealed flask as a result of meat putrefaction by anaerobic bacteria, a food spoilage process. The figure shows an illustration of Francisco Redi experiment. The first flask remained open, the middle sealed, while the third flask covered with a piece of gauze. We see that in the opened flask, maggots appeared, formation of larvae on the meat, while the sealed, no larvae, the third also no larvae, but the sealed one in the middle, a food spoilage process occurred, a putrefaction of the meat. In order to protect food from microbial spoilage, Spallanzani suggested heating the food extract to kill food microorganisms as a food preservation process, and the diagram illustrates Spallanzani experiment. When he heated the broth or food extract and left the flask open, wait for a time, growth appeared, while when he sealed the flask, no growth appeared. After being opened, the flask will be contaminated. English scientist Tyndall didn't agree with Spallanzani because the heat didn't kill all food microorganisms. His idea is the raw food usually contaminated with two groups of microorganisms: the food contamination sources, heat labile vegetative cells, and heat resistant bacterial spores. In order to overcome such a problem, Tyndall suggested the discontinuous heating in a process called Tyndallization of the food, in which food heated for 3 successive days, every short period of time, in order to promote the germination of bacterial spores which resist heating on the first or second day, and converted to vegetative cells on the third day to be sensitive to heat. After Tyndallization, the European scientists used oil bath, salt solution, steam pressure, autoclave in order to increase the temperature above the water boiling point to kill heat-resistant bacterial spores in a food sterilization process, and the figure shows autoclave. In 1795, Appert discovered food preservation by canning process, by placing meat in glass bottles and boiled it, and the figure shows an ancient glass bottle used in canning industry. In spite of all these experiments, the deep scientific works on food and industrial microbiology was made by French chemist Louis Pasteur from 1857 to 1887. Pasteur demonstrated that microorganisms are present in air and can contaminate food. So he proved that by filling several short neck flasks with beef extract and then boiled them, some were left open for a few days. The open flasks were found to be contaminated with microbes. The others, sealed or vented after boiling, were free of microorganisms. Pasteur placed the meat broth in a flask and bent the neck into an S shape or curve flask called the swan neck flask, and closed its opening with a cotton plug in the lowest point of the neck. The contents of this flask were boiled, and the meat content didn't decay even after months. He concluded that the cotton plug trapped airborne microorganisms to protect food from microbial spoilage. So another work is swan neck flask to prove that airborne microorganisms are a source of contamination. Pasteur made heavy works on food spoilage, on fermentation. He stated that raw food material contained many microorganisms, causing the spoilage of food product. He found in the French wine rod-shaped microorganism in addition to the oval-shaped yeast used for production of the product. So, rod lactobacilli contaminating the grape produced lactic acid instead of alcohol and spoiled the wine. The figure is from his observations. To kill road bacteria causing spoilage of wine, Pasteur suggested the pasteurization of the product. The same pasteurization process we used it now to pasteurize milk. Heavy works on all types of fermentation. Pasteur proposed germ theory of fermentation, that for each fermentation there is a special microorganism cause it. Based on the study of butyric acid fermentation caused by anaerobic bacteria Clostridium butyricum, Pasteur made his famous historical statement for the first time: life without oxygen. In addition to his work on food and industrial microbiology, Pasteur strongly proved that no living things created from non-living matter, and the spontaneous generation theory is wrong. Parallel to Pasteur's work, other groups of scientists made an emphasis on the role of microorganisms, food microorganisms, in the foodborne diseases and food poisonings, while the third groups focused on the beneficial phase of microorganisms or food microorganisms and used these microorganisms as a pure bacterial starters to produce improved foods. As the scientists Storch in 1885, who used for the first time a pure starter to produce Danish butter, and Friedrich Meissner in 1891 to produce Swiss cheese.